Hello class and welcome to the second video on the digestive system physiology. We will be taking a close look at the small intestine and its relationship to the liver and the pancreas, and how these three organs work together to accomplish the task of digestion. Let's first look at the gross anatomy of these organs, and then we'll take a closer look at their microscopic anatomy. The small intestine is a tube that is about 20 feet long in the relaxed state, and that equates approximately to the height of a two-story building. Now that is an impressive length, and there's a really important reason why the small intestine has to be this long. Now this picture is demonstrating the relative lengths of each of the components of the digestive tube, and we can see how the small intestine makes up the majority of its total length. The small intestine is the site for the majority of the chemical digestion and absorption that takes place, and it is the most crucial organ for these processes. As a matter of fact, we could live with the removal of any other part of the digestive tube, and people do, but we would not survive without a small intestine. Why don't we go ahead and name the parts of the small intestine, beginning with the part that extends from the pylorus of the stomach, and this is the duodenum. The duodenum is the busiest part of the small intestine because the majority of chemical digestion occurs here, and a lot of the absorption of those nutrients that have just been digested as well. Now, although the duodenum is a very busy place, it's the shortest part of the small intestine. The next part, the jejunum, is quite a bit longer. And then the last part, the ileum, is the longest part. And the ileum joins up with the large intestine at the cecum. By the time chyme reaches the ileum, most of the breakdown products of digestion have been absorbed. So technically, we could live without an ileum if we had to as well. We will spend the rest of this video talking about the duodenum and the processes that are going on there, and spend some time with a couple of friends of the duodenum, the liver and the pancreas. This picture displays very nicely the relationship between these three organs. The duodenum is the site where digestion occurs, but the duodenum does not produce the key players of digestion. That is up to the liver and the pancreas. So there needs to be a way for these two vital organs to transport their secretions to the duodenum, and this is done through a system of ducts. Bile leaving the liver does so through the left and right hepatic ducts, and will either be stored in the gallbladder or go directly to the duodenum through the bile duct. Pancreatic secretions will leave through pancreatic ducts, and the main pancreatic duct will meet up with the bile duct at the hepatopancreatic ampulla. So now that we know how these very important secretions get to the duodenum, let's take a look at the microscopic anatomy of these three organs. If we look at the histology of the small intestine, we can see the four layers that are consistent with the histology of a hollow organ. The mucosa is specialized for the job of absorption, and so let's discuss how that is so. There are four features of the small intestine that give it its incredible ability to absorb huge quantities of nutrients, vitamins, and minerals, and a surface area the size of a single tennis court. The first feature that lends to this large surface area is the length of the tube. The second feature is the circular folds of the mucosa and the submucosa. The third feature is a modification of the mucosa called the villi. And the fourth feature are the microvilli on the absorptive epithelial cells. And we will examine three of these features a bit more closely. We will be able to see two of these features very nicely on this picture. We can see here the circular folds, which I will be tracing in yellow. The circular folds incorporate both the mucosa and submucosa and function to slow the movement of chyme by causing it to move along in a spiral motion path, giving it a much greater time for contact with the mucosa. This increases the absorptive effectiveness of the epithelial cells. The villi are finger-like projections of the mucosa that increase surface area, and I'm tracing these in blue. Now I want to look more closely at the features of the mucosa and the villi. The epithelium is a simple columnar epithelium with a combination of absorptive and goblet cells. And between the villi, we see pits that contain intestinal tubular glands, and these glands secrete mainly intestinal juice, which is a watery mucus. Scattered throughout the intestinal glands, we see enteroendocrine cells that secrete two main hormones, secretin and cholecystokinin, which we will discuss shortly. Within the core of the villus is a dense capillary bed and a wide lymphatic capillary called a lacteal, and these vessels are very important in the absorption of nutrients. Within the mucosa, we'll also see mucosa-associated lymphatic tissue, and as we get further into the ileum, it becomes more organized into structures called Peyer's patches. Within the submucosa of the duodenum, we find numerous glands that secrete an alkaline mucus, and these are called duodenal glands. This secretion is key in neutralizing the acidic chyme entering the duodenum from the stomach. Normally, intestinal juice is slightly basic, ranging from 7.4 to 7.8, and it doesn't contain any digestive enzymes. Let's look even more closely now at an absorptive simple columnar cell. And we can see on its apical surface densely packed microvilli that has a fuzzy appearance under the microscopic view. This is how it gets its name, the brush border. The apical plasma membranes of these cells bear enzymes that complete the digestion of carbohydrates and proteins and are appropriately named brush border enzymes. That's going to do it for the histology of the small intestine. So let's now move on to the liver. The liver is an incredible organ with many vital functions, and it's one of the most fascinating organs to learn about. To name just a few of its functions, the liver removes nutrients from the bloodstream for processing. It's the major detoxification organ. It's the site of gluconeogenesis, which restores glucose levels from non-carbohydrate sources. It produces most of the clotting factors that we learned about with the blood. It produces the albumin, which is the main plasma protein. It produces alpha and beta globulins, which are essential for the transport of lipids and fat-soluble vitamins. And its main digestive function is the production of bile, which is needed to aid the digestion of fats. So as you can see, the liver is a very busy organ. Most of these jobs are the work of the main liver cell called the hepatocyte. The parenchyma of the liver is highly organized into lobules that have a very structured appearance to them. Each lobule is roughly hexagonal in shape with a central vein at its core. Within the stroma around the periphery, we see three vessels placed at each of the corners of the hexagon. The hepatocytes are arranged closely packed in plates, and sinusoids run within the spaces between the cells. This is quite a busy picture to get a clear understanding of what's going on, so I'm going to do a more simplistic sketch for you. You may want to do the drawing on your own as well as a study aid. You can always pause me if you want to draw along, and I will just stay here waiting for you. So this is how things work in the liver. Blood that is carried to the liver through the hepatic portal vein, which is coming from the digestive system, will be distributed to the lobules by the portal venules. And the blood then enters the leaky sinusoids. The hepatocytes will process the blood as it moves past them through the sinusoids, and all the sinusoids within a lobule will eventually drain into the central vein. And the central vein of each lobule will drain into the hepatic veins, which will leave the liver and drain into the inferior vena cava. Now, the portal arteriole is bringing oxygenated blood from the hepatic arteries to nourish the hepatocytes. At the side opposite of the sinusoids, the hepatocytes are releasing bile into bile canaliculi, which drain into the bile ductules and then eventually into the hepatic ducts to head toward the duodenum. And we can see that the bile moves in the opposite direction to the flow of the blood. So that's a brief introduction to the liver and its workhorse, the hepatocyte. Let's move on to the pancreas. As you learned in A&P1, the pancreas has both endocrine as well as digestive functions. It is actually two glands in one. It's an endocrine gland as well as an exocrine gland. For now, we will focus on the exocrine gland that produces digestive enzymes. Now, without the pancreas, we would not be able to digest most foods. So be good to this organ, as well as your liver. If we look at the pancreas under a microscope, we'll find typical looking exocrine glands, so the histology is not all that fascinating. The digestive enzymes of the pancreas catalyze hydrolysis reactions for all four macromolecules: proteins, carbohydrates, lipids, and nucleic acids. The enzymes that catalyze the breakdown of proteins, called the proteases, are in their inactive forms and are referred to as zymogens. These enzymes become activated once they enter the duodenum, and it's the brush border enzyme called enteropeptidase that gets this process started. The other digestive enzymes produced by the pancreas are amylase, lipase, and nuclease. In lab and lecture, we will be looking at the process of digestion further, and we'll be discussing these enzymes in greater detail. There's one more thing that I want to talk about in this video, and I need to go back to the two hormones that I mentioned that are released by the endocrine cells of the intestinal glands, and those are secretin and cholecystokinin. Both secretin and cholecystokinin will be released in response to chyme entering the duodenum from the stomach. And both of these hormones will stimulate the pancreas and the liver to increase their output of secretions. And that's going to do it for this video. We'll continue this discussion in class. Okay, so I'll see you then.